All right, let's dive into Kaposi sarcoma-associated herpesvirus. You know, for us in medical microbiology, really getting a handle on its unique biology is, well, it's everything. It's the only way to truly understand its clinical punch, especially right here in our local context. So let's get started. So what are we dealing with here? At its core, this is a gamma herpes virus. And that's not just a classification, it's a huge clue. This is a really complex pathogen, and its entire game plan, how it sticks around and how it causes disease, boils down to this fascinating dual life cycle. It's absolutely key to everything that follows. Just so you know, all the information we're about to walk through was put together using a Gemini deep search. If you're the type who likes to dig into the primary literature, and I know many of you are, you can find a full list of all the sources in the document linked below. Okay, so here's the plan for today. We'll start with what makes this virus a unique viral oncologist. Then we'll get into its incredible latent-lytic life cycle. From there, we'll look at the spectrum of clinical diseases it can cause, dive into treatment from a Dutch perspective, and finally, wrap up with diagnostics and prevention. All right, Section 1, a unique viral oncologist. So what exactly is this virus, and what's its place in the world? You have to remember, its discovery back in 1994 was a huge deal. It was a landmark moment, really, because it was the missing piece of the puzzle that finally explained those devastating cancers that became such a tragic hallmark of the AIDS epidemic. So we're talking about Human Herpesvirus 8. This is the cause, the etiological agent of all forms of Kaposi sarcoma. And here's the central theme you're going to hear me come back to again and again. Its power, its ability to cause disease, is almost completely tied to the host's immune status. That's the critical variable. Now, let's zoom in on its family tree. It belongs to the rhadinovirus genus, and get this, it's the only one we know of that infects humans. This isn't just trivia, it's super important. Why? Because rhadinoviruses are notorious for something called molecular piracy. Think about it. They literally steal and repurpose our own genes to make our cells grow uncontrollably and at the same time become invisible to our immune system. It's a brilliant, if sinister, strategy. Okay, now for the main event, really. The latent-lytic lifecycle. This is the core of its pathology, this two-part strategy that drives both its persistence and the diseases it causes. Let's break it down. Okay, so if you look at the variant itself, it's got that classic herpes virus structure. No surprises there. But the real magic, the crucial part, is what happens to its genome once it gets inside a host cell. The linear DNA actually closes up into a circle, forming what's called an episome, basically a tiny little extra chromosome that just hangs out in the nucleus. And here's the genius part. A viral protein called LANA acts like a molecular anchor, tethering that viral episome to our own host chromosomes. So every single time that cell divides, the virus gets a free ride into the daughter cells. It's an incredible persistence mechanism. And that persistence mechanism is what allows for its two-state strategy. Think of it like this. Latency is the default mode. It's the virus sleeping, hiding out as that episome, expressing very few genes, just trying to stay off the immune system's radar. But then, when the host is under stress, say from immunosuppression, the virus wakes up. It flips the switch to the lytic cycle. And that's an all-out production phase. It starts replicating its genome, building new virus particles, and ultimately it bursts the cell open to spread to new cells. Now, this is where it gets really interesting, because the cancer isn't caused by one cycle or the other. It's the synergy between them. Latency is the engine of the tumor itself. It makes the infected cells proliferate, building the tumor mass. But a tumor needs a supportive neighborhood to grow, right? That's where the lytic cycle comes in. A few cells will sporadically reactivate, and when they do, they spew out this cocktail of viral proteins and hijacked host factors, things like VEGF and IL-6. This creates this perfect inflammatory pro-angiogenic microenvironment that feeds and sustains the whole tumor. It's a truly vicious, self-sustaining loop. Okay, so we've got this pathogenic mechanism down, but what does it actually look like in the clinic? Well, the consequences of this dysregulated life cycle are incredibly diverse, leading to a whole spectrum of clinical disease. Yeah, it's really important to understand that Kaposi sarcoma is not just one thing. It shows up in 5 distinct epidemiological forms. You've got the classic type, which is pretty slow-growing in older men. Then there's the endemic African form, which can be very aggressive in kids. Iatrogenic, which we see in transplant patients on immunosuppressants. And of course, the epidemic AIDS-associated form, the most aggressive of all. And now we even recognize a 5th subtype in HIV-negative men who have sex with men. The thread connecting them all, the patient's immune status. And it doesn't stop with Kaposi sarcoma. The virus is also behind a couple of really nasty B-cell lymphoproliferative disorders. The first is primary effusion lymphoma, or PEL. It's this rare, super aggressive lymphoma that causes these malignant fluid buildups in body cavities. And the other is a specific form of multicentric Castleman disease. This is a massive systemic inflammatory problem, basically a cytokine storm that's being directly fueled by lytically infected B cells going haywire. So, that brings us to the big question. How do we treat this stuff? Let's talk management, specifically focusing on the Dutch perspective, moving from immune reconstitution all the way to local control. You know, for cases where the disease is limited and not really progressing, especially with the classic form, the approach here in the Netherlands is often what we call Afwachtend beleid. It literally means a wait-and-see policy, or watchful waiting. We don't jump in with treatment unless the lesions start causing problems or clearly begin to spread.
This really breaks down the strategy nicely. Look, rule number 1, the absolute foundation of any treatment plan, especially for anyone who's immunocompromised, is to restore immune function. So for an AIDS patient, that means getting them on combined antiretroviral therapy, or CART, immediately. That's paramount. Now, if the disease is limited and localized, we can use local therapies, things like cryotherapy or radiotherapy. It's only when the disease becomes progressive or widespread that we bring out the big guns, systemic chemotherapy. And the first-line choice, both here in the Netherlands and by international consensus, is liposomal doxorubicin. Now, treating kids is a whole different ballgame. It's particularly challenging. For one thing, the presentation can be totally different. You might see severe lymphadenopathy, this major swelling of the lymph nodes, without any of the classic skin lesions you'd expect. And here's the really crucial point. There is a huge research gap. We just don't have enough pediatric-specific staging systems or clinical trial data. So a lot of the time, we're forced to adapt treatment regimens from adult protocols, which is, well, it's far from ideal.
Okay, let's wrap up with diagnostics and prevention. How do we identify the virus and stop it from spreading? Well, transmission is mainly through saliva, but also sexual contact and, importantly, organ transplants. So prevention is really about standard precautions, promoting safe sex, and screening organ donors, particularly in areas where the virus is common. So how do we actually find it in the lab? This is a key point for any microbiologist. You can't just grow this virus in the lab like you would with many others. It is notoriously difficult to culture. People have tried on all sorts of cell lines, and you just don't get that classic cytopathic effect. It just doesn't work. And that's why quantitative real-time PCR is the absolute, undisputed gold standard for diagnostics. We usually aim for the ORF73 gene. Remember, that's the gene for the LANA protein, the one anchoring the episome. Since LANA is expressed in every single infected cell, whether it's latent or lytic, it's the perfect target. And this is so important. Measuring the viral load isn't just a yes or no for diagnosis. It has real prognostic power. If you have an at-risk patient and you see their viral loads start to climb, that's a huge red flag. It can actually predict that they're about to develop the disease. And on the flip side, when someone's on treatment, watching that viral load drop is one of our best markers to know if the therapy is actually working. So this naturally leads to a really critical question, one that keeps us up at night with other herpes viruses like CMV. Should we be worried about this virus developing drug resistance? And the answer is, well, it's basically no, but not in the way you might think. It's not about genetic mutations. The problem is that our antivirals, like anciclovir, can only attack the virus when it's actively replicating during the lytic cycle. But the virus spends most of its life asleep, in that latent state. So it's not genetically resistant, it's biologically resistant. It's hiding where the drugs can't see it. And this is exactly why we don't have standardized susceptibility testing for it. The real challenge isn't a mutated polymerase, it's the biology of latency itself. And that really leaves us with the final big picture question, doesn't it? If we want to truly cure these cancers, the ultimate challenge isn't just about finding a better chemotherapy or a more potent antivirus for the active virus. The real frontier is figuring out how to get rid of that silent, persistent, latent reservoir. So I'll leave you with this thought. How do you kill a sleeping virus? I really hope this deep dive has been useful for your work. If you enjoyed this, please subscribe for more explainers focused on clinical virology. We genuinely value the expertise in this community, so please leave your feedback, your thoughts, and any questions you have in the comments below. Thanks for watching.